All right, we want to thank ZONTAL for sponsoring the delicious lunch during this presentation. And at this point, I'll introduce Christof Gänzler, who's the Director of Pre-Sales and Product Marketing for ZONTAL as well.
Thank you very much, and thank you for showing up. This is like the very slowly going towards the end of the show. So I'm happy that you're here. Hope you enjoy my talk today. And we are ZONTAL. Whoever doesn't know us, we are turning data into corporate assets, your data. And we have the idea of storing, preserving all of your lab data, whatever flows through your lab, whatever is produced in your lab is stored inside ZONTAL, it's annotated inside ZONTAL, it's preserved inside ZONTAL to really create a fair data platform. So I will quickly introduce ZONTAL and then show you what you can build, what we can build, what our customers can build with the entire platform. And this is one of the steps towards a real digital lab. So my dear end-to-end analytical request workflow that everybody would like to have is, well, for a chemist should answer the question, did I really make what I planned to make in my little reaction tube? And this workflow can be very automated, very easily, if you have the right tools. And the benefits of such an automation are very clear. We're reducing human error. And nobody likes to take notes about every little tiny thing that we're doing. So let's let the engines do it. Let the systems do it for you. And ultimately allow all of these data be shared and used for further analysis. And this is the R in FAIR that occurs automatically with ZONTAL, and I will try to show you what that means. And in a typical project goal is that, hey, we have too many systems accessing our instruments. And when something changes, all of these individual connections have to be redone. Or only 3 of them, and for like 2 others can stay. But to find that out, it's it's always the same process. You have to go through, you have to revise it, you have to reprogram it, and so on. So the idea is that we looked at the broader issue with lab data in general. And yes, a very similar picture to the one before, but a little bit more detailed. So we have the expert data analysis tools that every scientist needs to make sense of the data. The ELNs to plan what we actually do in the lab, LIMS to execute it. And specialty data platforms, they're everywhere. And then we have data scientists. They should be everywhere. Not everyone has enough, I know. So every scientist needs to be a little bit of data scientist as well, which answers my question to one of yesterday's panel discussion is it easier to teach a programmer biology or to teach a biologist programming? And I think it's easier I can explain biology much better to a programmer, so it's probably that way around. But yeah, in the end you also have, and this is a big thing for every company out there, you would like to archive your data. You would like to make sure that you can actually, after 5 years, still find that data and still be able to interpret it. Not only view the data, but really do something with it. So what we built at ZONTAL is a fair data layer. And that means that we are connecting all of these processes, all of these workflows, and we're connecting the instruments, we're connecting the solutions, the ELNs, the LIMS of the world, and we're connecting them with the direct output of the experiment. And that doesn't only go one way. We are orchestrating the workflows. We really try whenever we can, take data like a request, an instrument request, a sample request, whatever, a test request from an ELN system. We take it, we interpret it, we change it so that the instrument that this request is for actually understands it. We send it to the instrument. So when the scientist goes to the instrument with the samples, with her samples, then the sample list is already there. They can put in their plate or their vials and push the start button to test. ZONTAL is there waiting for the next step. The next step is the instrument produced output files or some output that we wait for. And as soon as this output is produced, this is picked up by ZONTAL. We ingest the data, we parse the data, we put it in a data package that actually contains all of the metadata and all of the data flow. So we're annotating this data and we create one package out of that. And of course the results are sent back to where the planning was done in an experiment, so that this is there again in the system where it came from, or in another system, it depends. And so if it's really well done, you don't even see ZONTAL. And I will show you a couple of examples today where these integrations with the software to the instrument and back are really seamless. And for the scientist, sometimes they see ZONTAL because it makes sense for them to have a visualization in between in their workflow to make sure the quality is the right thing, or to have it completely in their normal tool, and they don't even recognize that there is an entire horizontal data layer in there. So let me introduce this operations workflow, and this is one example that was initiated by a customer a couple of years ago who said, okay, we want to have a scientist being able to do a test request, push a button in the request system, walk to the instrument, do the test, and by the time the scientist goes back to her desk, the completely analyzed data must be already there. And this is how it ended up as being the really streamlined workflow for lab operations. And as I said, this is an example of us working together. You see the red dots here with our sister company MasterLab, but this can be any other analysis tool. So this is more in the chemistry space, but you're creating, you're planning your reactions, you're executing your reactions, you're measuring them. You get back not only the results from the instrument, but you get back the metadata as well. And of course the planning data, so the structure of the desired structure. ZONTAL takes these results and sends them in the right format directly into MNOVA to number crunch it, to do the first analysis of, hey, is that what we got from the NMR in this case, is that actually representing the structure that we put in first, that we have planned for? And of course, we get that also back from MNOVA, and we put, again, everything into that data package. So we have the planning data, we have the execution data, we have the results as files in this case, because the NMR spits out this nice list of files. And we have the MNOVA results And we have a nice report because Nova spits out a nice PDF report as well. And everything is in one, so people can look at that inside ZONTAL, or they can look at it because we send it back into the ELN system, into the experiment where it came from. So let me take you quickly through a couple of screenshots here. If somebody wants to see it live, no problem. I'm here, or just contact me and we can do a live demo. But this is Signals Notebook. In this case, can be any other ELN system. So I'm a molecular biologist. Please, the chemists, forgive me my not very correct chemistry here. But this request goes into the system, and ZONTAL, I show here ZONTAL because, of course, this is all available. All of this information is available inside the ZONTAL system. What we do here is we actually send it to the NMR. The NMR gives us back, in this case, NMR produces also a PDF, so that's why I put it here. So, but we also get the raw files from the NMR machine. We send these raw files into MNOVA to say, okay, here is the structure that originally came from the ELN. Here's the result that came just from the NMR machine. And please use this script to say, okay, is this structure actually represented in this result? And there are just a couple of steps necessary in this workflow inside ZONTAL. And this is becoming, more and more customers, the standard way of doing these repetitive workflows. Because the scientists say, oh, we don't want to change how we're working, and that's totally fine. It's still the same workflow for them. What we automate is we are reducing the amount of manual drag and drop and copy and paste and interactions to an absolute minimum. We are creating an entire workflow that has an entire audit trail. We are orchestrating the back and forth, including, if you wanted to, the method. If we're talking mass spec, we even sent already the method with the entire package, with the samples and with the instructions to the machine. This was work that we did with the Pistoia Alliance. So all of this is one data package. All of this is one information workflow, and it can be tracked and traced for every scientist, and the scientists are, in this case, are very happy because they don't have to search for the right files on the instrument PC to copy them on a shared drive. So this is what they have, what's in it for them. But of course, on a higher level, what you get is a lot of data integrity. It is faster, as I said, 2 minutes between the read and it appearing back in the notebook. And what you get is a full audit trail of every single step that was carried out in this workflow. And by the way, this is true for everything that ZONTAL does. The audit trail is there. For every ontology term that you're using, for every setup that you're doing, everywhere there's an audit trail. Even when you delete and purge files out of ZONTAL, this is a record in the audit trail, so the audit trail won't go away just because you're deleting the file. Okay, so just for this first part of the talk, really the idea that we can have here biology systems, all kinds of chemical systems, standalone systems, whatever, your scales, your pH meters, and we're connecting this to the systems that are used by the scientists, or that they have to use, like SAP. So we're ingesting and normalizing that data. We're preserving it, but we're also making it available for further analysis. So you can go across all of your NMR spectra that were produced and in the system and see, okay, with which instrument were they produced over time, in which labs, which software was used to analyze them, things like that. Questions that you can now answer with this ZONTAL. And as I said, we would like to transfer the manual processes to automated processes. That's the true goal. We're doing this by having a fair data platform that cares about the data orchestration and the data integrity. And ultimately what you get out is a platform that you can use really to train models to make sure that every new data point is coming in is actually piped through a model and already said, okay, this is a good quality, this is a bad quality result, whatever. So these things are possible because all of the data is there. And another example with MasterLab in this case, since we had this nice connection already, we said, okay, the end user said, but I'm a power user, I need my MNOVA. I need this tool to actually look at this data, but like individually, not automatically. So what we do have, and this is together with MNOVA, we have MNOVA Hub that of course can connect to your normal drive, but it also can connect to ZONTAL, and ZONTAL looks like a hard drive to MNOVA. So it's no change for the scientist. They just use it, they open the MNOVA Hub, they see all of their results from ZONTAL, and they can just click on it, open these results. This is again an NMR, yes, and work with it, and even save it back and create a new version of that information package that I was talking about, because now they have analyzed the data, so they're adding just another layer to it. And it is, of course, like searchable content, and it's like literally clicking on it, loading it as if it would be a normal file. But it's not only a normal file. What the scientists are creating here is, of course, a full analysis of that NMR result in this case. And then we save it back into ZONTAL. Other people can again see it. It is connected to the original run, is connected to the original experiment. It is connected to including all of the metadata, even to the point where it's connected to the ontology, so that you can even search for it in a couple years' time. And the interesting piece, and it's very small here, and I don't expect you to read that, the interesting piece here happens without the scientist even knowing it. By saving it back into ZONTAL, MNOVA creates a set of metadata. Like, if we have a structure in MNOVA, this structure will get a UPAC name, and this name is stored in ZONTAL. So you can actually search in ZONTAL for that name, and you will find all of the MNOVA files that contain this structure. And many other chemical properties are saved by default into the metadata set of ZONTAL. So we're not only saving the file, we're saving much more of the content of the file as metadata, which is all the time then accessible, and you can again do your analysis with this data either inside ZONTAL, because we can of course search and sort and visualize the data inside ZONTAL, so we have an NMR viewer, so you don't need the MNOVA necessarily, the MNOVA software. So this is all one package, and this is just really to illustrate this here. This can be done with any other software as well. So when you have information that not necessarily needs to be in all systems, but you need your system of record, ZONTAL in this case, to have this information, to know there is a certain gene name in an analysis. There's a compound identifier that is stored in another system. We pull that in, and this is all adding to the metadata of that file. So, and with that, I hope I didn't bore you to death, and we can have some questions, and I'm happy to take them.
So thank you. That was very nice, and it's a nice looking product. I'm over here. Oh, big guy right here. I really like the way that you've got everything all pipelined together and all the scientific instruments connected to the data and all that. My question pertains to the fact that in many cases instruments or the computers running them are not registered in scientific databases. They're registered with facilities and are in a completely different sort of format. They may link out to the owners of those instruments or the owners of those files, which would be registered in a user directory run by HR, for example. And I'm wondering, do you address those types of issues?
Yes. We have, that's a short answer. One sentence more to this. What we do while we're pulling in every data set, we are annotating this data set, and this link to this HR system or to the system of the manufacturer would also be queried. So we're putting this in. It's a very simple script that we can make extremely powerful if you want to. Our standard is literally 4 steps, but yeah, if you need more, we're pulling all of this data based on file names, based on like information inside the files. Yeah, sometimes in the file name is actually contains information that we need for annotation, and so on. So this is what we're doing when we're collecting these information packages.
Question. The instruments you're connecting with, right? Does it require a headless API? The reason I ask that, if I'm using an instrument connected to an external PC that's driving the instrument, it might have access control. There might be things that you then have to get around, right? That's part one of my question. Part two is, you talked about, you have input into the instrument, right? It's going to have some, let's say I'm running an assay, right? And it has an XML file to define all the parameters. And then you're going to get something out, which is the run data, right? And then there has to be some term grounding, because they might not, what you put in and you get back has to match in some way. And not all instrument manufacturers are going to make that available, or they're not going to have maybe mapped it. So one time they might say current and the input, and then the output says amperage or something, right? So yes. Those are my 2 questions.
Yep, we are in constant exchange with the manufacturers about that. And usually it's customer driven. So we have over 400 instruments that we can read data from already. We can send 2 instruments directly, and that's part of this integration that we have. Not all of them allow this. We can go through third-party tools. That's what we're doing if we have like a lab execution system that knows how to do that. We're going through that, or a LIMS system. So these are the things that we can do during the workflow. Work with these other systems to get it in, and honestly, sometimes it's just the customer sitting down together with the vendor and us and define how to do that. Most of the times it's possible, to be honest. Welcome. So yeah, if you have a special question, just come to me later.
Just a quick question. Thank you for the very interesting presentation. And I'm not trying to put you on the spot with the question, but the value proposition of ZONTAL kind of reminded me a little bit of our annual sponsor here at TetraScience. I don't know if you get this question often, but from your perspective, how would you differentiate the offering, and what is it that you would want to highlight for your customers that they would want to know?
I wouldn't even try to differentiate. Yes, we read data from instruments, we're pulling data in, that's fine. We're also pulling data from TetraScience, because that is like, that's where people have data. But usually what we are doing is go directly to the instruments and read the data. Because this everybody wants the whole track record of what has happened to this piece of data that comes from there, because it did not, like this data record there on the instrument is only there because somebody was planning it in a different tool to actually exercise it, execute it, and then run this instrument. So we have the full workflow mapped inside ZONTAL, and we have this aspect of data preservation also with the original files. So everything is in this allotrope, usually in the allotrope standard file, where we capture the metadata, the data cubes, and the original files all in one system. And this belongs to the customer. They can take this file and store it somewhere forever. The good thing is you can take ZONTAL out of the equation. When you have this allotrope file, you can rebuild the entire system if you wanted to, because the information is all encapsulated in one information package. You're welcome.
All right, any other questions?
Thank you very much.